Okay. Thank you again for joining us today for a tutorial entitled as Decoding Rhizosphere Dynamics using molecular and imaging approaches. So I just have a few slides as introduction. So actually the fantastic talks today in the two sessions that we had kind of set the stage for this tutorial very nicely and made my job kind of easier on why this is important and why we're gonna do this. As you know, the rhizosphere is the most complex microbial habitat on the earth, comprising an integrated network of plant roots, soil, and a diverse microbial consortium of bacteria and other microorganisms. So understanding, predicting, and controlling the structure and function of the rhizosphere will allow us to harness plant microbe interactions and other rhizosphere activities as a means to increase or restore plant ecosystem productivity and mitigate effects of climate change by designing ecosystems for long-term soil carbon storage. So also this type of studies will lead to developing rhizospheres and microbial sensors, which can be used to design, test, and validate molecular sensors related to microbial functions and root exudates for continuous monitoring of chemical and nutrient exchanges in the rhizosphere. So EMSL offers several capabilities and expertise in this area. And we are really, happy to work with users to actually use these capabilities, employ these capabilities in this type of research. Today, under this tutorial, which we're just going to cover only a portion of this of the capabilities that EMSL could offer in this area. Of course, there are many others that can be achieved in EMSL website, and you can always, get back to us and ask for any questions. So our presenters today are Dušan Veličković, a mass spectrometry imaging scientist at EMSL, Jim Moran, chemist and lead scientist. He's also lead scientist for ecosystem interface integrated research platform at EMSL, and Curtis Larimer, a material scientist. So I also want to go through the agenda very briefly. So after this short introduction, so Dušan will take it. We'll be mainly focusing on the use of multi-spec imaging for spatial omics in your environmental research. And then, so after each session, we have its own Q&A. So everyone are encouraged to ask questions. You can even write it in the chat box, or if you want, you can unmute yourself and ask your questions. And then after first part of the tutorial, then Jim will take that. And then there are 3 back-to-back sessions from him. But again, after each session, we can pause and have that Q&A session for each. And then the last part of the tutorial will be by Curtis, which will be mainly focusing on optical coherence tomography or ACT imaging of plant roots. So we hope that this tutorial provide some very good fundamental information for current users or even future users to start working with us. Great. I think I can unshare this, and Dušan, you can just take it from now.
Yeah,
thank you.
Sounds good. You just need to stop sharing.
Yeah, just let me stop share.
Okay. Probably. This better? Do you see?
Yeah, looks great.
Can you see? Okay. Yeah. Thanks, Amir, for introduction, and thank you all for joining this tutorial. So this part of tutorial will be actually dedicated to probably the most employed molecular mass spectrometry imaging tool at EMSL, and it is matrix assist laser desorption ionization, or MALDI, mass spectrometry imaging. So I will give you really brief fundamentals of this technique, but I want actually to emphasize where and how we can help you and how we use actually this tool in environmental research with examples from different EMSL user projects. So okay, so MALDI mass spectrometry imaging actually, as other mass spectrometry imaging techniques, provides insight into distribution and identity of different kind of molecules, so primary, secondary metabolites, lipids, glycans, even proteins. And what is important, it does in untargeted fashion. So you don't need to know anything about chemistry of the sample before you analyze your sample. So you don't need to use any antibodies and specific targets. It is completely untargeted approach. What MALDI provides actually in terms of spatial resolution, we talk about something 10, 15, 50 or 50 micron spatial resolution, so near single cell level. And that spatial resolution actually depends on how well we can focus our probe on the sample, how well we can focus laser on the probe. So if you look here, you can see that actually how mass spectrometry imaging works. We use the probe that is specifically focused on small areas. We call it pixel in the sample. And then we, in case of the MALDI, we use the laser. We ablate material from that localized spot in the sample, and that ablated material is ionized, actually molecules in that ablated material ionized and goes into the mass spectrometer, where we actually measure mass of those compounds. And then when entire sample is analyzed, so we took from each particular pixel in the sample mass spectrum, we actually end up with creating ion images that actually just represent relative distribution of particular molecule with particular mass over the sample that we analyze. And in this case, you can see how that looks like on the root tip. Okay, so in MALDI, mass spectrometry imaging, we have that word MALDI, so matrix assist laser desorption ionization. In MALDI, we actually apply a coat of the organic compounds that are called MALDI matrices to help in ionization of those endogenous molecules in the sample. So this following video will actually show you how we do it in a reproducible way and limited any batch effects that can occur from sample to sample. So Okay, so what I want from you to remember is that in mass spectrometry imaging, key is that we link chemical with spatial information. So any kind of application, as you saw this, for example, matrix that will enhance ionization of molecules can cause delocalization of the molecules. We want to avoid it, and it is the reason why we use those sophisticated devices that deliver matrix in the form of the time mist and minimize, actually prevent any kind of delocalization, so we can be really confident that where we see molecules in our images, it is where actually the molecule is present. So once when we coated samples with the multi-matrix, we can go ahead and analyze the sample, acquire mass spectrometry images, so acquire those pixel by pixel mass spectrum, and to do that we use at EMSL very actually ultra high mass resolving and ultra high accurate mass spectrometers that are currently available. So this video will give you a brief overview how we acquire our data. Okay, once data are acquired, we actually process those data. They're really enormous, huge files. You can imagine that you have several thousand of different mass spectrometry features from one pixel. And for processing those data and visualization of our images, we also use sophisticated softwares that enable us actually to see and see that relative distribution of molecules and the relative quantification and differences in abundance between different cell types or between different tissues that we analyze. So this short video will show you actually what kind of data we can generate. Okay, so now when we understand some basic principles of analyzing and using multi-mass spectrometry imaging and kind of data that it can provide, I want to actually share with you what kind of samples do we analyze at EMSL, and what type of scientific questions, environmental scientific questions, we can answer with this tool. So probably the most common sample that we analyze at EMSL are colonies that are grown on the solid agar plates. So by using mass spectrometry, multi-mass spectrometry imaging, we can actually basically analyze microbial communication and metabolic exchange between colonies on the agar plates, as in this case, for example, in communication between cyanobacteria, moss, and fungi. Also, we can go and analyze topography of microbial colonies, chemical topography of microbial colonies. We can actually analyze sagittal planes of colonies grown on solid agar, so we can take cross sections of those colonies and then have insight into distribution of metabolic processes that occur in the depths of the biofilm, and then reveal some phenotypic differences between different cells in the biofilm, between the different cells that have different biological function in the biofilm. Then we can also analyze, for example, interaction between roots and microbes, in this case, roots and fungi, in terms of, for example, mycorrhiza formation. So we can take, for example, longitudinal sections of the roots and then visualize metabolic translocation that occurs during compatible or, for example, incompatible interaction between fungi and the roots. Then we can also create, for example, 3D metabolic maps of plant tissues. In this case, it is soybean root models, so another association between plant and microbes. And what we actually do here, we take and analyze serial sections of soybean root models or any other plant organ, and then basically stack those 2D images to see basically 3D distribution of metabolic processes in the plant organs. Then we can also go beyond surface, go and analyze subsurface biochemistry. So what's going on in the rhizosphere or deep in the soil, and for this purpose we use techniques that calls indirect multi-mass spectrometry imaging. So we don't analyze directly soil or rhizosphere. We actually imprint, as you can see here on the left-hand side, we imprint either rhizobox or soil box, and then have insight about organic distribution around the roots, like in the left-hand side image, or where they are localized, how for example they are exudated in the soil, or if we do that a similar approach with the soil with the soil box, we can also have insight into, for example, different soil microbiome features in different, in this case was different humidity throughout the depth of the soil. Then we can also analyze how plant responds to environmental stress, especially stress that is especially on the localized response. In this case, for example, wounding, mechanical wounding of the leaf. We just take we just wound the leaf, for example, with the surgical clamps, and then take cross section of the leaf, and then can see actually where specific metabolites are located, and actually we can track if they're located exactly on the position where we wounded. So it can be a really good way to track, for example, signal transduction during plant response to some environmental stress. lastly, we can also follow, for example, plant biomass decomposition. And in this case, we use unorthodox samples like wood slices taken from those wood buffers where fungi was growing. And then we can actually have insight into the real time of lignocellulose degradation by brown rot. We basically, as you can see here in space, we can separate time of degradation, and then by analyzing different slices of this wood buffer, we can have insight of what lignocellulose degradation products are in which stage of degradation, or what fungi metabolites are in exactly same stage of lignocellulose degradation. And our multi-mass spectrometry imaging group is pretty big, so I would also to thanks to those folks that are also involved in this work. And yeah, I'm open for your questions.
Thank you very much, Dušan. That was wonderful. So now we can have questions for Dušan, if there are any. You can write it in the chat box, or you can just unmute yourself, turn on your video, and just talk.
Hey, Dušan, this is Adam Mangeli. I have a question.
Mm-hmm. Yeah, okay.
So for the Rhizobox application of the MALDI MS, how long does that I don't know what to call it, like the coating, how long does it need to be exposed to the roots to get a reliable measurement of what's actually there?
Yes, so in, yeah, good question, really. So in this particular example, I believe we did that. So we did that blot, I think 24 hours. It was like a blotting of this membrane to the Rhizobox to transfer molecules on it. But just note here that our coverage here was not like we were able maybe to see around 50 different compounds. So we still need to improve this in terms of changing chemistry of the membrane that we will transfer the material there. So we still hope that we can improve. But currently, I think that in this particular case, this blotting was around 24 hours. So and a good thing here is actually that you can do live monitoring. You can just place this membrane after 5 days, then remove it, then again after 10 days, and then monitor basically like how root is growing and what is changing with root exudates there.
Yeah, that's exactly what I was thinking about doing. So
yeah.
Thank you very much.
Thank you.
So I see one question in the chat box. So I might have missed it, but how long does it take to map the localization of the image in the first figure? With the ablation and MALDI.
First figure. Which one was first figure? I'm not sure. Okay, probably. Yeah, this one, right? I mean, this was the first one of application. So yeah, I mean, I can in general tell you. So approximately, it depends from spatial resolution that we use. It depends from mass resolution that we use. So you typically, for example, if we talk about this particular example, usually it's about let's say count that it's about one second per 1 pixel. So it depends like how many pixels you will have. I know that, for example, for like poplar root cross section, you need approximately 1 hour to analyze it. For this particular one, I'm not completely sure. I think around 5 hours you need to finish entire acquisition, but count like approximately 1 pixel per second, roughly like that. just to acquire data. After that, we need to process data as well. So just to see, to map the localization in the first figure. I mean, I hope I answer with this. Again, depends of the number of pixels you will have, but I can tell like approximately 1 second per one pixel.
Great, thank you so much. Any other questions for Dušan? So probably not, but as I said, you can always reach out to Dušan and all the other speakers. Yeah, I think that's it. Thank you very much.
Thank you,
Dušan, again. So I think with that, so Jim will take it from now. So as I said, I already put the agenda for this tutorial, a picture format of that in the chat box. So if you want to see what you expect to see in this tutorial. And then, so I think Jim is also a fan of even more direct live interactions. So if, so it's okay if you want to interrupt Jim while he's talking. Am I right, Jim?
Yeah, that is wonderful. What I'm not a fan of necessarily is technology. So you're gonna have to give me just a second while I try to shuffle screens around.
Yeah, but we will still keep that few minutes Q&A after each part of Jim's presentations, because he's actually covering 3 different topics. So we'll have a pause for Q&A between in each topic. Okay, thank you very much. Stage is yours.
I'm guessing you're seeing the wrong screen?
No, I think it's, that looks good to me.
Oh, good. Okay.
Yeah.
Well, let's see if I can even advance it. It goes to the next slide.
Yeah. Yeah.
Perfect. Okay. So you all have to apologize. My screen that's showing is not the screen with the camera. So I'll try to walk through these slides from a side view. And as Amir said, feel free to stop, interrupt me with any questions. If we don't get through all the slides, well, oh well, it's better not to leave any dangling questions there. The first slide I have here is not necessarily a technique we're going to talk about, but I wanted to put this up because I'm going to discuss a few different methods. And all of these methods have been developed using rhizoboxes, right? And just wanted to give a little bit of background information, how we developed the rhizoboxes, what's contained within them. And all of the soil that we've been using is from the Kellogg Biological Station in southwest Michigan. And it's all taken from some switchgrass plots that they have. Now let's see. So with that sort of background, I want to jump into some of the work that we've been doing trying to map enzyme activity and also looking at specifically proteomics analysis of rhizosphere. So here's the first example. I mentioned, the rhizobox. Here's an example of the rhizobox on the left. And then what we've been doing to attempt to map enzyme activity is this sort of blotting technique, right? So we take the side off the rhizobox, put a membrane up there, make some contact with the soil, and let it incubate there. The incubation time can be on the order of tens of minutes to, in some cases, even hours, or in a couple of cases, even a full day, even 24 hours. We don't want to go too much longer than that because at some point, we're using nitrocellulose membranes. At some point, that might be used as a carbon source, and we might start to really perturb the system and some of the microbial responses. But as long as we have it up there long enough, we can then peel that membrane off, take our rhizobox, put the cover back on, put it back in the growth chamber. The plant can keep growing. It's completely non-destructive, non-detrimental to the plant. But now we have this membrane, and there's a few different things that we can do with it. And one, I'm going to show you a couple examples today. The first is this top component of the greenish component of the figure. And that is, we can perform some level of enzyme assays on that membrane, right? What we're counting on is that enzymes were attached to the membrane, we peeled it off, they retain their activity, we can leverage that activity to locate where the enzymes are and sort of map that. And the second is that we can also stain what's on that membrane and identify what the protein distribution looks like. Where are the proteins, and specifically, where are they in relation to different types of rhizosphere? So here's one example of that. This is a nice sort of autofluorescence image of a root system in the rhizobox. In that rectangle, blue rectangle, we've applied a membrane. And then we've looked at both the phosphatase mapping distribution as well as the total protein distribution. And you can see probably a pattern that you might expect, right? At the root tips, you're seeing a little bit more phosphatase and even proteins, probably from some of the exudation and sloughing of cells as that root grows. And what we're able to do is leverage these plots, as I have here, for looking at phosphatase and also a proteomics analysis. When we're looking at these enzyme activities, we can map where they're located. And again, this is a very non-destructive technique. So here's one example. I should say that when we're mapping, we're using, in this case, a phosphate sort of surrogate. Really, it's DDAO phosphate, which is a fluorescent dye effectively with a phosphate group on it. And when a phosphatase cleaves that terminal phosphate group, the dye then becomes two things. It becomes one, fluorescent, and two, it becomes insoluble in water. So on this membrane, you apply the phosphatase surrogate. When a phosphatase enzyme acts upon it, it precipitates on that membrane, becomes immobile, also becomes visible for fluorescent imaging techniques. And that's what we've done in this case. Here you have root 1 and root 2. Root 1, we have, you might see within the red circles, some organic phosphorus sources. That's really ground up plant material from a previous experiment. The soil, I wouldn't say is marginal, but it's certainly not replete in phosphorus necessarily. What we wanted to do is ask the question, do we see a spatially focused increase in phosphatase activity at a location that is close both to the root and also close to the organic phosphorus source? Not a super thorough experiment. I don't want to get hung up on that, but I do want to use this data to exemplify what we think we can do with the technique. In the middle plot, you can see some of the results, right? You can see that root, and within these red circles, it looks like, at least anecdotally, there's an increase in the response of this phosphatase mapping in the close proximity of that organic phosphorus resource. And then on the right side of the slide, you can see that when we compare based purely on pixel intensity, mathematically computing the pixel intensity within a certain distance of the root, we can see that we do seem to have a larger intensity of phosphatase activity near root 1, which goes next to the organic phosphorus material, compared to root 2, which goes next to an inorganic non-nutrient sort of control. And I should mention, right now we have the phosphatase capability. We can roughly adapt that for an acidic and alkaline phosphatase assay. We're also working on chitinase activity using a very, very similar, pretty much the identical approach with a different stain. But we want to look at enzyme activity. We also wanted to try to be able to enable proteomics, but spatially resolved proteomics, right? We all know how important different spatial constraints can be when you're looking at rhizosphere. And we recognize that all of the rhizosphere along a root is not necessarily the same, right? You have a whole bunch of different stages as a root is pioneering a new soil, developing new rhizosphere, or further up the root where there might be a little bit less exchange and interactions between the soil and the plant itself. So to do that, we turn to this type of approach. Hopefully you can see my cursor, but a same type of sample, again, a plant in a rhizobox in soil. We image, just visually image the root, and then apply a membrane. It can be the same membrane. We actually use nitrocellulose membrane, the exact same that we use for the enzyme mapping. Fix those proteins on the membrane. If we pull the membrane off, fix the proteins on the membrane, stain them. And once we stain them, we can get something like what you see here. You can see the root, and towards the tip of the root, there's quite a bit more response from the stain. It's staining quite a bit darker. We then go in literally with a biopsy tool, take this very thin membrane and cut out pieces that we're interested in. In this case, you can see that we've really focused, really targeted our approach on just the proteins that we can extract from really the root tip versus a more mature rhizosphere up the distance of the root. We've taken these membrane pieces, we do a digestion of the peptide, of the proteins to peptides right on that membrane piece, and then we can apply Orbitrap mass spectrometry to perform proteomics. Here's an example. We had this simple question: can we distinguish the root tips from the mid-root proteome? You know, looking at rhizosphere, very nascent rhizosphere versus more established rhizosphere further up the root. You can see we've applied a few different membrane pieces in this case, trying to encompass sort of a vertical cross-section over the root. And you can see also on the far right panel, the spots at the bottom where we selectively sampled either the root tip or more mature rhizosphere samples further up the root. And what the results look like is something like this. When you, well, first of all, we observed 6 biological orders that contain, you know, the vast majority of the proteins that we identified. And really, we saw a difference in the taxonomic distribution of these proteins in these 2 spatial locations. So at the root tip, we're seeing some bacterial representation, but a lot more of the Liliopsida, the switchgrass, right? A lot more proteins from switchgrass at that root tip than at the mid-root. However, we're seeing more bacterial taxonomic diversity represented in the proteomics of that mid-root, sort of further up the plant root. This also, you know, if we pull out just the switchgrass data and look only at the bacterial data, you see a very similar trend. We're seeing more bacterial-associated proteins, higher abundances, higher representation of them in the proteomics data from the mid-root versus the root tip, and also seeing potentially a bit more taxonomic diversity, more bacteria being represented in that mid-root. And we're hoping that this is what you might expect, right? As a root is pioneering its way through the soil, basically it's potentially moving faster than the bacteria can keep up, and it takes a little while to develop that more full community behind the growing root tip. Here's just another example, the same type of data. You can just see that the dark blue switchgrass proteins are statistically much more abundant, much more preferred in the root tip than the more mature sections of root. And that is in contrast to a few of the other bacterial orders, which have representatives that are more abundant and seem to prefer, if you will, the more mature mid-root section of the samples. So we also, you know, I have quite a background in stable isotope analysis. We wanted to begin seeing if we could couple proteomics analyses with stable isotope analysis. And so this is a slightly different example. In this case, we're doing a little bit more of bulk proteomics, but we're doing it in an interesting sort of rhizobox, a little bit of a switch from what I told you before. Here we have a mature switchgrass plant, and we're really performing a split root experiment. So on the left, we have the root biomass is in some soil. There is Kellogg Biological Station soil in here, but also quite a bit of sand, which we've used to, in effect, almost dilute the nutrients, if you will. We applied Hoagland solution on the left side of that split root rhizobox to really improve and increase the nutrient abundance. And on the right side, we also applied Hoagland solution, but Hoagland solution devoid of phosphorus. We wanted to ask the question that, are we seeing a response, a localized response in the split side of the root system that doesn't have phosphorus, or at least a response based on phosphorus availability. So I will say that we observed 3,000 proteins when we did this, and that some of these, about 700 of them, did show a significant preference, again, if you will, preference in quotation marks, for one of these 2 sets of conditions. What didn't appear to show a preference, however, is really the root exudation. We might have, just only looking at the bulk, the amount of root exudation using a 13C tracer, we saw nearly equivalent amounts of root exudation in both sides of the rhizobox. But if you dig into those proteomic results a little bit, you'll see that there was not the same distribution, not the same taxonomic distribution of proteins in each case. We were seeing more proteins related to switchgrass or attributable to switchgrass in the high phosphorus condition than the low phosphorus condition. But there's also a number of different bacterial proteins that appeared to have a more dominant abundance in one side or the other. Specifically, if we looked at the microbial responses, there were almost 200 proteins that had preferential abundance in the high protein, in the high phosphorus, and then another 200 in the lower phosphorus conditions, and much more preference, I would say, for the switchgrass proteins in the high phosphorus condition. We wanted to go beyond just that. You will recall that I mentioned we used a 13C tracer. EMSL has really been spearheading this effort to try to be able to automate in an automated fashion, pull out proteins that contain a 13C label. And this is in general how it works. When you incorporate enough 13C tracer into a protein, or into a peptide in this case, you begin to see an enlargement of the mass spectra, sort of a blurring of it over into higher masses. So sort of these mass ranges to the right would indicate that this peptide has potentially some extra 13C attributable to the tracer, and that would not naturally be there. So we performed analysis of the proteins that we identified to say, okay, which of these proteins contains that 13C tracer? And what we found is that in each case, roughly 12% of the total proteins extracted from both halves of the split root system contained proteins with that 13C tracer. So we're getting nearly equal amounts of labeling on both sides, which is consistent with this very similar rates of root exudation that we saw on both sides. But what is different is that we're seeing these proteins being associated with slightly different taxonomic groups, right? So obviously we're seeing a strong switchgrass presence, but also a strong switchgrass presence for 13C labeled proteins in the no phosphorus side, but some differences as well in some of the bacterial taxonomy associated with these 13C tracers. So again, we're not seeing a whole scale difference in the amount of root exudation in both sides of this rhizobox. We're not seeing a wholesale difference in the percentage of proteins that are incorporating this tracer, but we are beginning to see a difference in especially the bacterial taxonomic distribution of these 13C tracers into the protein component that we're looking at. And we're trying to track that down a little bit more. Another thing that we're trying to develop is what I have up on the screen now. And we're hoping to couple this also with the 13C tracer work. And what we've started doing is taking very small physical samples of the rhizosphere soil right up next to a root. This is really enabled by some of the really microfluidic proteomics processing capabilities that have been developed at P&L and specifically EMSL. And I can't stress enough the small amounts, tens of milligrams of soil, something like that. By taking such a small sample, it's giving us the ability to really spatially target where that sample comes from. It's not a non-destructive technique, right? We're actually physically removing soil, we're altering the system, but we're removing a very tiny bit of soil, such a small amount that you could consider this approach conducive to time series sampling, right? Especially if you're willing to consider a time series where you sample in one location, and the next time point you might sample in 2 or 3 millimeters away from that along the route. Again, we're targeting rhizosphere with the full recognition that not all rhizosphere along a route is identical, right? We don't want to just isolate all the soil and do, bulk rhizosphere proteomics, because we may be blurring a lot of spatially driven heterogeneity in the process. Now, one of the great things about this technique is that we're getting very high yields. So looking at the mid-root and root tip, very similar to what we did before, but you're seeing that with our quote-unquote new approach, the direct soil approach, we're getting a higher number of proteins that we're able to identify compared to the old. And we're also seeing really the expected proteomic diversity as you look at this. So we're probably getting a protein bias at some point, but we're also getting a large enough sample set that the bias is not dominating everything that we're seeing. So here we're seeing a much higher diversity, for instance, in rhizosphere soil versus bog soil further away, still within the same rhizobox, but further away from the root. And what I should also mention is that by getting deeper coverage of the proteomics, despite being, somewhat a little bit destructive, we're still getting a deeper coverage, and I think that's going to really enable a stronger science, enable us to address questions that we might not be able to address in other ways. The other really fascinating thing about this, we're still working out some of the final components of the method. One of the great things about it is that I think this can really be accessible for EMSL users. So if you have Rhizobox or maybe even field systems, if basically all you need to do is harvest that tiny amount of soil, put it in a sterile tube, freeze it, and send it to us, and we could perform this type of analysis with it. So as Amir said, we're going to talk about a few different things that we've developed here, but I did just sort of cover up the slides I had for the proteomics analyses and enzyme mapping. So I wanted to break very quickly and see if there were any questions at this point. And I can't see the chat window. There we go.
I don't see a question in the chat box, but I have one question, Jim, for you.
Sure.
I think that was slide 7 or 8 when you were talking about a different trend that you see with the plant proteins and the specific microbial community proteins in different region of the root. So root tip shows kind of different trend than the middle of the root.
Yeah.
So my question is that, have you tried different developmental stages of plant, or that was just one specific developmental stage? Or you think if you try other stages, maybe you see a different trend?
Yeah, this was all from one developmental stage. I will say that we did multiple rhizoboxes. So it's not just, there were, I can't remember, 4 or 5 different rhizoboxes where we pulled the data. But it was all really close to a single stage. But I think, Amir, you hit on one of the great things. We could, because this is non-destructive, this could enable time series analyses. Yeah. So certainly you could capture a couple of things. You could capture different stages, or you could capture one point in soil before a root gets to it, when a root is hitting it, and then as a root passes by, and you could watch really the evolution of the microbial taxonomy at that one point.
Yeah, that would be very cool. And also, maybe I'm wrong, but it should be some ways to start trying to correlate the nature of the plant proteins with the particular microbial community, and try to see which possible functional, or functions can be proposed for why we have these particular proteins with this particular microbial community in that particular region of root, in that particular developmental stage.
Yes. Yep. Exactly.
Yeah.
And I will caution. There's one caution here, in that, as we saw with some of the later data, this really spatially resolved, non-destructive approach does get a little bit less protein coverage in the results. Yeah. So fewer numbers. So I don't know if we'd want to construct networks, interaction networks, or things like that necessarily. Just the one caveat, right, of the data.
Yeah, but this is really wonderful. I do see one question.
Yes. Yeah, I'm reading the question now. Yeah, so the question really is, where are the proteins from? Bacteria, plant, expired bacteria, what is the current thought? I think the answer is yes, really, in a way, right? Most likely these proteins, with the membrane sampling approach, the proteins have to be at least soluble enough to get to the membrane. So they're probably not coming, we're not sampling the proteins from the roots, say, but we would be sampling proteins that might be exuded by the roots, right? So number of different proteins might be exuded from the plant roots, or exoproteins would be exuded by bacteria. Also, certainly lysed bacterial cells could be another a source of these proteins. All of these, I think, could definitely be a part of it. Okay.
Wonderful.
Now, let's see if there are no more questions, and feel free to interrupt if there are. I'm going to go on to the next section very quickly. I don't know how I'm doing on time, but hopefully we're okay.
I think you're good. Yeah.
Good. Okay. So we want to track root exudates through 13C tracers. Now, there's a very different goal here, right? So what Dušan mentioned was really sort of tracking what are, almost performing metabolite analysis. What are the root exudates? Things like that. Can we identify them chemically? What we're trying to do here is subtly different. We're trying to, instead of identify specific chemical compositions, we're trying to quantify how much root exudate is occurring and where is it occurring. Okay, so slightly different approach, different target, different goal. And for all of this, we're using a 13C tracer, where in the slides that you're going to see, the tracer is really applied through a 13C CO2 label that's applied aerially to the plant. It's incorporated through photosynthesis, and then the resulting root exudates are transported below ground and exuded into rhizosphere. So we know, for instance, little quick refresher, I'm sure you're all very familiar with this, that root exudates fuel a number of subsurface biogeochemical processes. A lot of soils are carbon limited from a microbial perspective. Plants can help remediate that, and certainly rhizosphere is one of, if not the biggest source of subsurface organic carbon in a soil system. So root exudates can provide an important carbon source for different microorganisms, and there's a lot of different compounds that can go into these root exudates. Maybe some of the root exudates aren't even intended for microbes, but they're meant to do a biogeochemical function on their own. But we also know that the time and location of these root exudates can really vary over growth cycle, seasonal cycle, a growth stage, things like that. So our goal here is to develop a method to track plant-derived carbon into the rhizosphere and into soil, and both track it and potentially also quantify it. So there we go. I mentioned that we're using a 13C tracer for all of these. Here's a depiction of a growth chamber that we've developed to apply 13C tracers to the leaf area of plants. And then the first method I want to talk about is a laser ablation method. So we're coupling laser ablation to enable spatially resolved sampling to an isotope ratio mass spectrometer, which enables high quality quantification of the amount of 13C. Again, we're using this 13C tracer as a proxy for root exudation, right? When we apply the tracer to the headspace, we're allowing it to get taken up into the plant, transported to the subsurface, and released. And if we can track where that 13C goes, we're going to link that directly to how much root exudation occurred in that location. Now, what you see here is sort of a bait and switch, I'm sorry, but this is actually a hair. And you can see in the hair, there's three holes clearly drilled in it. The reason I have this here is really to provide some sort of frame of reference for the spatial resolution that we can get with the laser ablation approach. Each of these hairs is about 50 microns in diameter, and we're able to harvest enough carbon from each of those holes to make multiple 13C measurements. So what I'm going to do now is take you into the lab and see if we can get into a little bit more detail about how we make those measurements. And everyone, please yell at me if the volume's not loud enough or for some reason this doesn't come through.
Jim, it's not coming through. So if you want to pause it real quick.
Yep.
And just go up to your Zoom, controls up at the top of your screen.
Yeah. And I'm going to go to more, correct?
And then click share sound.
And we'll try it.
Get laser ablation isotope ratio mass spectrometry.
Good.
And what this capability is it's a way to get spatially resolved 13C measurements along roots or rhizosphere or even soil interfaces. Here's a sample that we've already used. You can clearly see the root going down the middle of the sample. What's harder to see are the small ablation pits on either side of the root. This is where we've already taken a number of samples for precise 13C measurement. One of the nice features of this is that while it's a very destructive activity, a destructive sampling, that destruction is very spatially focused. So we have plenty of material left here for other types of analysis. What's also very hard to see is that we have complete control over where we want to place these ablation pits. We tend to place them about 200 microns apart, but we could place them closer together if we wanted to. And we can align them so that we're clearly looking at the decrease in 13C that we observe as you get further and further away from the root itself. Finally, I want to mention that there's really a minimal amount of sample prep involved. We grew this plant in a rhizobox. To do the sampling, we took the side off the rhizobox.
So I can't, did it get stuck?
Yeah, it looks like it stopped
Effectively
playing.
took a very thin soil core from that rhizobox going in.
Has it already played this? Sorry, I can't hear the sound on my end. Is it working?
Yeah, it's
in this case, the z-axis of the sample to flatten out the surface. But this retains the two-dimensional spatial resolution of the sample. So we haven't altered the two-dimensional spatial components here. We then dried the sample in a freeze dryer, and that's it. Once we have our sample prepared, we can simply place it into the laser ablation chamber, close it up, slide it into the unit, close the front door, and then we're ready to visually image our sample and select the subsampling locations. The CO2 generated from the combustion reactor passes through this capillary. When we place this capillary in liquid nitrogen, it cryotraps all that CO2 into a very small area of the capillary. Once we've trapped enough CO2, we can remove the capillary from the trap, and as it warms, the CO2 sublimates away, is carried by the helium carrier gas through the rest of the line into the isotope ratio mass spectrometer where we make our measurement. What the laser ablation system allows us to do is target a specific location within a sample that we want to take a subsample. In this case, I'll put a target right here. We can then turn the laser on, and what it's going to do is flash a number of pulses right at the surface of that sample in that targeted location. That's going to remove little small pieces of that surface of the sample. We're going to entrain that in helium, combust any organic carbon in that sample to CO2, and then pass it to an isotope ratio mass spectrometer for highly precise quantification of how much 13C is in that sample. Let's take a closer look. So now we've looked at the basics and the high-level overview of laser ablation isotope ratio mass spectrometry. But I want to stress again what we can do with that data. This data can provide us spatially resolved analysis of where things like root exudate are going within rhizosphere systems, which can help us elucidate different hotspots of activity, monitor where they are located and how long they may persist in the rhizosphere.
Okay, I'm hoping that's done, so I'll go forward. Hopefully that provided a little bit of an introduction to how we make the measurements. Now I want to look a little bit at what some of the results look like. So here is a root sample. We've harvested out of one of these rhizoboxes that I mentioned we'd be using. And when we perform the laser ablation IRMS on it, you can see here that the color is depicting sort of the range of 13C values we get. So we know, for instance, that root A is receiving a lot more fresh photosynthate. It has a lot more 13C tracer, suggesting that there's a lot more of that fresh photosynthate coming from CO2 to root A than there is to root C, for instance. Whoops. And then also, sorry. And then similarly, root B is getting a lot more carbon than root D, for instance. We're not trying to assess why this might be occurring, but we think we have a technique that allows us to determine which root is being more emphasized in the fresh photosynthesis, the fresh flow of carbon from the plant. And I just put this up here. Because of the quantitative nature of the isotope ratio mass spectrometer, we can also quantify these differences between roots. Here, looking at the differences between root A, B, C, and D, for instance. Here's another sort of pictorial example of this. You can see the root here, and then the different laser ablation pits, sort of perpendicular to the root. And when we take the data from those points right there, we get something that looks like this, right? We see the root has the most 13C tracer. We can clearly see in this 13C space the rhizosphere sort of bridging the root on either side. And we can use this approach to look at how thick is that rhizosphere? How much carbon is going to the rhizosphere in different sections of the root, or when the root is put into different conditions. Now, this is really great. I think we're getting on the order of probably 75 to 100 micron spatial resolution when we do this. But I also want you to understand that EMSL is always trying to push the limit, right? We're trying to increase the number of samples that we can take, the number of data points we can take, increase the spatial resolution. So to that end, we're also working to develop a new technique. This is actually a spectroscopy versus a mass spectrometry approach, leveraging some hollow fiber capillary absorption spectroscopy approach. We actually have a patent on this at EMSL. So we're using the same laser ablation approach, but coupling it to a spectroscopy instrument measurement platform. Here's a picture of our laser that we're using for that, combustion reactor. Then on this piece of actually cardboard is a small capillary, very similar to what you might think of for a GC, gas kept chromatography system. When we put CO2 in there, we can clearly see transitions, see features in the spectroscopy related to a 12C and 13C component of that CO2. And we can use that to measure the amount of 13C in each of the samples. So here we have an example where we have a root. These large pits are composed of samples that we took for laser ablation IRMS in the sort of dash box. Now, the improved sensitivity of our spectroscopy platform enables us to take smaller samples. And so in this solid rectangle here, you might be able to squint and see some of the ablation pits that are noticeably smaller in size because we needed to harvest less material from the sample. Yet despite that, we see very similar trends. So the open circles are the results we get from the IRMS, mass spec approach. You can see, the root is the most labeled, and then sort of going away from the root on either side, you're seeing a decrease in the amount of label, the amount of tracer. But the black dots are what we're seeing with the spectroscopy approach. Very similar type of sample, the identical sample, shifted slightly in space. Sorry about this. But what I've tried to do is scale the size of the points to reflect the size of the ablation pits, right? So with mass spectrometry, we have these huge ablation pits. With the spectroscopy, we have much smaller ablation pits because we get better spatial resolution. And one of the interesting things that we're beginning to see is that a lot of times when we apply the spectroscopy, we're seeing this decrease in the amount of label right in the middle of the root. And we think this is, I don't want to get into the sort of the geometry too much of the root architecture, but we think what we're seeing here is we're able to pick up the different components of the root tissues where we're actually seeing an increase signal in sort of the phloem component of the roots that's transporting the fresh photosynthate down into the subsurface. And we're not seeing quite as much enrichment, maybe closer to the middle or in areas that are not transporting that labeled photosynthate. So I think that is the last slide I have at this point then with tracking the 13C tracers. I can pause briefly if there's any questions with that. And if not, I can go right ahead. So what you've seen is we're trying to acknowledge that there's spatial components of rhizosphere are crucial, right? There's so much heterogeneity in soil and rhizosphere. We're trying to develop ways to pull that apart. You've seen our approaches at trying to spatially map enzymatic processes and their distribution in this rhizosphere. We've tried to pull out proteins that we can link them to microbial taxonomy and track shifts in the microbial taxonomy in rhizosphere in different spatial locations. We've just looked at looking at a different supply of root exudates to different sections of rhizosphere as well. This final section that I have for you, we're really trying to look at, can we look at the inorganic nutrient environment in different sections of rhizosphere? To do that, we're turning to another laser approach. And you might think that we're laser happy. This one's a little bit different than the laser ablation. And this is called laser-induced breakdown spectroscopy. And what this approach does is you take a laser beam and you shoot that laser beam right on the surface of the sample. What happens when that beam hits is it delivers a lot of energy in a very small spatial location. It creates a near instantaneous plasma, right? So you superheat that area. The electrons in that area associated with different atoms get excited, and then as they relax back down to the ground state, you can perform spectroscopy through that plasma, and that can give you some indication of what kind of nutrients, what kind of elements you might have there. And that's what you see on the right. You can see, for example, this calcium line is really tall. We're getting a lot of calcium in this case in the soil. You can also see that we're getting a large number of different elements that we can identify in this matter. We've looked at just a few of the ones that we can get. You can make these measurements every time you make that plasma. You can scan through these different wavelengths and really get a collection of different a collection of information on different nutrients, different elemental nutrients in the soil. And so here's an example of what this looks like, same soil, and we've just highlighted here with different colors some of the different, barium, manganese, magnesium, silicon, even carbon in some cases. And these really are consistent. I don't want to say that we're measuring the mineralogy, but they're consistent with some of the feldspars, the quartz in some cases, carbonates. They're very consistent with what we know exists in the soil. We can also apply these to roots. So I apologize if this is small on your computer screen, but in this upper left box, you can see a couple roots. And then moving to the right of them, you can see that where the roots are, when we apply the LIBS approach, you can see really the carbon in that root, those roots really shines, right? Also, to some degree, the hydrogen depicting the organic content of those roots. But more interesting, we can also look and effectively map different macronutrients, micronutrients, and then what we've termed here soil matrix components. So silicon, for example, or aluminum in some cases. Now we can pull together data from these maps and begin to piece together trends, so spatial trends that we're seeing through the rhizosphere. So consider on this plot, the x-axis 0 is at the edge of the root. As you're moving away from the root, you see an increase in some elements, right? An increase in potassium, phosphorus, iron, calcium. And we believe that's depicting this depression of these concentrations near the root where these ions have been consumed and brought up into the root, into the plant biomass. On the other hand, as you progress away from the root, you're seeing a decrease in the amount of carbon and hydrogen. Now, this is very similar with the trends we saw before, where you're seeing root exudation, where there's more root exudates up near the root, and that concentration decreases the further away you get from that root edge. And the last thing I want to mention on this LIBS approach is just that the sample prep is fairly minimal on this. So really, we've taken our rhizobox, we've effectively just drilled out a little mini core on the side of that, and then we press that to flatten the surface. We need a fairly flat surface to do that spectroscopy there. Then we dry it, and that's it. And then we can go from that sort of pellet that we get and put that right in the LIBS device and perform the analysis. So that is the last slide I had, and I want to thank you. And then I can break one more time if there might be any questions out there. Okay, sounds good. Thank you.
Thank you very much, Jim. That was really wonderful. Okay, so yeah, seems like there's no question. We can definitely move forward with our next speaker. Curtis, please take it away.
Yeah, hi, Amir. Can you hear me okay?
Yes.
And the slide came up?
Yeah.
Great, okay. So hi, everyone. I'm Curtis Larimer. I'm a material scientist. What I'm going to talk about in the next few minutes is just a relatively straightforward non-destructive imaging capability that we have, and that would be available to you all. It is an optical coherence tomography instrument. So first, what is OCT? It's a tomographic microscope. It's often referred to as a kind of optical ultrasound, right? So it uses the same mechanism and produces images that honestly look a lot like, you know, an ultrasound you might have of, you know, a baby, right? But instead of sound, it uses a low coherence near infrared laser light source. It's really non-invasive, non-destructive, and what you can get out of it is really nice 2D and 3D imaging. As you can see, that's a picture of the actual instrument on the right there. It's a relatively small footprint, just easy to sit on a bench top. It's pretty easy to move around, so if you have a sample that's, you know, already set up in EMSL in one particular lab, we can usually bring this imaging system to you. And we've even done things like taking the imaging end, which is on the right side, and fish that through a port into a growth chamber or something like that. Okay, so what's OCT good for? You know, it's great for doing three-dimensional surface or even subsurface imaging. You can image deeper if your sample is transparent and it's less scattering. So in a root, you'll be able to image deeper than you would in surrounding soil, because the soil is made up of a lot of scattering particulates. That root's also, you know, probably going to be, you know, white or, you know, partially transparent, which will help light penetrate deeper. What you get out is a 3D image where you can then cross-section through the depth. So you can kind of see that in those images on the right where there's a seed. And just I've cut cross-sections across from left to right and then from front to back so that you can kind of see the first few cell layers there. Like I mentioned, this is pretty fast and it's non-destructive. So you can do time series. And we found that, you know, you can take images over days or longer, just depends on how frequently you want to image. It's only really limited by how much hard disk space you have available on that computer. These 3D tomographic images can get up into the couple gigabytes per image of file size, and so if you think about doing hundreds or thousands of them, it gets big pretty quick. In terms of the limitations, if you want to try to image below 1 to 3 millimeters, it really has to be an idealized, you know, mostly transparent sample. In the kind of plant and root samples that we did, 1 to 3 millimeters is pretty much the max. In terms of lateral resolution, it's not great, you know, pretty good, but not great. 4 to 12 microns. That's just a function of using that infrared, longer wavelength infrared light. In the axial resolution, that's your Z direction or your depth resolution. It's 4 to 6 microns. Field of view is pretty good, you know, 6 to 16 millimeters, so you can see, you know, pretty wide area, a whole leaf or a whole, definitely a whole root. And a really nice, good long working distance. So if you don't want to get too close and disturb your sample, you can do it from, you know, 3 to 40 millimeters away. This imaging technique does not work if you have thin reflective surfaces. So like a window that's made from a cover glass would not work. But we can and do image through windows as long as they're thick. That just has to do with there being many light reflections between really thin stuff that kind of messes up the imaging. So I just took this little graph showing on the y-axis how deep you can image with different techniques, and on the x-axis how much resolution you can get in each of those techniques. And you can see that OCT kind of stretches up to that upper left corner more than other techniques, in that you can get more depth and resolution than some comparable techniques. All right, so let's show you how we actually use it. This is one of Jim's rhizoboxes. It has a switchgrass plant growing in there, and a bunch of nice roots growing down through the soil. So what we did was we turned it at an angle so that the roots would grow right up against the window. Like I mentioned, imaging really deep into the soil is not easy with this, but you could do it as long as it's kind of at the surface. And then the imaging arm is actually on the left and kind of pointed up to the right along that 30-degree angle. And then we just use some foil to cover the box so the light in the growth chamber doesn't interfere with the root development. So here's some examples of the images that we've taken. On the left side is like the 3D image. It's shown in perspective, and you can see sort of from the left going right across the middle, right above the black arrow is a root. And taking that same image and tilting it so you're looking at the edge, you can just see the end of that root and even sort of some of the cell structure around the sides of it visible in the side of the image. So I'm going to show some videos of this because, like I mentioned, we can do time series, and I think the videos are pretty cool. So there's no sound with these. I'll try to explain what's going on. This was the root growing in soil, and this is over about 9 hours. You can even see it's probably maybe just too small to see on your screen, but there's just a very fine set of root hairs that start growing out from the side of the root. And there's enough good resolution to see those. So take a look for the white arrow in the middle, and you can see a root hair just above that, as well as 3 or 4 more just behind the one that's indicated. I'll let it go one more time. So we're watering the plant while we're doing this, and it's obviously alive and growing. And sometimes there's a little bit of trial and error in finding the right route to image. We probably took 3 or 4 different series like this until we got one that really showed the movement like that. Okay, so obviously once we've got a series of images, we can do cool things.
Sorry,
like kind of
Curtis?
Yeah.
So is that is it how many days or hours of imaging?
Yeah. Oh, right. That series was over about 9 hours with taking images every 15 minutes.
Oh.
But we've definitely done imaging series that go over much longer. You don't have to be sitting at the instrument. You can pre-program it to take an image at regular intervals. And so if you want to do every 30 minutes or every 15 minutes, it can run all night while you're sleeping, or you just check it again in the morning. There's a little bit of a risk, right, that something gets out of focus or something, but we didn't encounter that too badly.
Great. And is there a need for a specific software to extract the roots, or the images, or it will be just straightforward and right away you can just see?
This instrument comes with its own software for operation, and that's what generated the 3D images. But you can export it in a number of pretty common formats. So if you already have techniques to work with 3D images, then I'm sure that we can get the data from this instrument into a format that you can use.
Great.
Yeah.
Thank you.
And it doesn't have to be a 3D image like this. You could also set it up to do a line, and then you just get one profile. That can be advantageous because the full 3D image, it might take 30 seconds or so to take the field of view that you're seeing on this slide, but a single line can be done, in many times a fraction of a second. It's actually imaging at, I think it's 20 kilohertz or something. So you can do things like image while you're moving the imaging end around, and you can still have pretty sharp images. Okay, so what was I saying? Once we've got this time series, we can see the root growing through the field of view. We can track in this case, I just tracked the tip of the root and where it moved. That's the black line in the image on the right. And then used just some other software to really straightforward measure the distances and the times and how fast it was moving. So we measured, the growth rate in millimeters per hour for the main root and for the root hairs, and we found they're pretty comparable, which is interesting. Okay, another video, and this is just to show that we don't have to just do roots, we don't have to just do stuff in soil. If you have samples where you want to look at leaves, or you want to look at stems or seeds, I think the seeds image are pretty cool. You can see a lot of the fine detail of these structures coming out, and all of that's imaging in air is easier than imaging in soil. So that's all available as well. Okay, so I think that's all I had. I'll open it up for questions. And while we're answering questions, I'll just show one more video because this one's pretty neat too.
Yeah, very cool videos and very nice applications for that.
Yeah, so
lots of potentials.
We've definitely talked about, like with Jim's work, right? He wants to take these really high resolution measurements, and he wants to take and make a laser ablation spot just at the right time to capture a specific phenomenon. So you can envision using this technique to track the growth of that root until just when it comes into contact with a specific nutrient in the soil, a nutrient island, and then take that measurement right when it gets there, take it as it grows past, and so forth. So that's what we see it as. It's just a nice non-destructive way to monitor for things. And then, of course, it also just takes nice, good pictures that can go into publications and whatnot to provide some visual support to other measurements that are done.
Awesome. Yeah, I also see lots of potential as using this to complement many other capabilities that we have, and as you just mentioned one example to work with you.
Yeah.
So any questions for Curtis?
I have a question, Amir.
Chat window.
Yeah, go ahead.
Go ahead.
Hold on.
So I'm gonna admit that I had to step away from my computer and I missed the majority of your talk. And I would really appreciate it if you could just explain kind of the depth of penetration on this one more time, and kind of how these data are collected. And again, I apologize, but everybody's doing 10 things at once. So.
Exactly. Yeah. No worries at all.
Thank you very much.
As a reminder, and I'll try to jump back to that slide. The imaging depth is 1 to 3 millimeters. It depends a lot on what kind of sample you have. So if your sample is, really scattering, like a bunch of little particulates, it's going to be a lot less than 1 millimeter. If your sample is pretty transparent, pretty smooth, you could go even deeper than 3 millimeters. I think that the manufacturer says you can do 5 or 6 millimeters kind of maximum. Okay, so in terms of resolution, well, you can see the numbers there. If you have more specific questions about how you might want to use it in your sample, just feel free to reach out to me. In terms of how it's collected, it's a really simple imaging arm, and you just place the sample underneath of it. Do a little bit of work to tune and focus, set up your scanning area. And then once you have it set, then it's 30 seconds to a couple of minutes to collect the 3D tomographic image, depending on how many line scans you're doing and how wide the field of view is.
Thank you very much, Curtis. I think I share the same opinion with a lot of people that are seeing this right now, and they're really impressed with the images. And that's exactly why I'm interested in using some of this stuff. Is we're working on some geophysical techniques to kind of monitor this stuff, and coupling that more blurry geophysical image that we get with some of these higher resolution images will be really beneficial, I think. So thank you very much for doubling back and covering that for me.
No, great, no problem at all. Just feel free to reach out to me, or if you have Amir's contact, he knows where to find me.
Sure, thank you.
Okay, great. So I don't see more questions in the chat box. What I'm going to do, I just copy paste the EMSL instrumentation webpage in the chat box. As I mentioned earlier in this tutorial, that what I covered today, what we covered today was just a portion what EMSL PNNL can offer to do works in the rhizosphere region and studying rhizosphere dynamics, but there are many more capabilities that can be used for that purpose. Please feel free to explore that website that I just put the link. And I would like to thank all the speakers again today. Dušan, Jim, and Curtis, all I think did a great job, wonderful. And please reach out to the speakers anytime if you have any additional questions. Thank you very much.